welcome to the deep dive. Have you ever stopped and wondered, why some infections suddenly get really hard to treat, even with our best antibiotics?
Or how, say, a bug like E. coli, which usually just lives in our gut, can sometimes turn into something really dangerous.
Exactly. And these aren't just random events. They're fundamental shifts happening right down at the level of the microbe's blueprint. We're talking about changes in microbial genetics.
It's absolutely fascinating and, honestly, pretty critical stuff. Antibiotic resistance is a perfect example, something we see unfolding constantly.
Like with MRSA.
MRSA, yeah, and even scarier things like multi-drug-resistant bacteria in hospitals. Think Acinetobacter baumannii, it can actually pick up resistance genes from other bacteria around it.
Wow.
It just shows how incredibly adaptable these microorganisms are. It's evolution happening right in front of us.
It really is. So that's our focus for this deep dive, delving into that hidden world of microbial genetics.
Yeah.
We're using Microbiology, an introduction, 13th edition, as our main guide here.
Good source. Very comprehensive.
Our mission today is really to unpack how microbes inherit traits, how they adapt, how they evolve. We want to give you those key nuggets of knowledge.
Right, the essentials. From the absolute basics, the code of life itself, all the way to how bacteria become these superbugs.
And hopefully without making you feel like you're cramming for an exam.
You'll try. And it really does matter to you, the listener, because understanding these genetic mechanisms is crucial for tackling things like emerging diseases.
You mentioned E. coli O157.H7 earlier.
Yeah, a classic case. It wasn't always so harmful. It actually acquired the genes for a powerful toxin from another microbe through, well, gene transfer.
And it's not just about disease, is it? Genetics helps us study microbes we can't even grow in the lab.
Absolutely. Sequencing allows us to peek into the lives of these unculturable organisms, learning about their roles in the environment, even within our own bodies, our microbiome. It opens up whole new worlds.
So let's start at the beginning. How do we even talk about this genetic information? The cell's genome, what does that actually mean?
Think of the genome as the cell's complete library of instructions, everything it needs to know.
The whole manual.
Pretty much. It includes the main operating system, which is the chromosome. That's the big structure carrying most of the hereditary info, packed as DNA.
Okay.
And often, there are also smaller extra bits of DNA called plasmids. Think of them as optional software packages carrying extra features.
And the actual instructions on these are the genes.
Exactly. Genes are specific segments of that DNA, or sometimes RNA, in viruses that code for functional products. Usually proteins, but sometimes things like ribosomal RNA or transfer RNA too.
And DNA itself.
Yeah.
It's famous for that double helix shape.
Right. Two strands twisted around each other. Each strand is made of repeating units, nucleotides.
And the key is how the bases pair up, isn't it? Adenine with thymine.
A with T, and cytosine with guanine, C with G. They're complementary. One strand perfectly matches the other.
And that's not just neat, it's fundamental.
Oh, absolutely fundamental. First, that sequence of bases, A, T, C, G, is how the information is stored. It's like a four-letter alphabet writing out the instructions.
It's so compact.
Incredibly. And second, that complementarity is why DNA can be copied so accurately when a cell divides. Each strand acts as a template for making a new partner strand.
Precise duplication.
Extremely precise. It ensures the instructions get passed on reliably.
Which brings us neatly to what Francis Crick famously called the central dogma.
Ah, yes, back in '56. The flow of genetic information. DNA makes RNA, and RNA makes protein.
DNA to RNA to protein. That's the core pathway for how the genetic message actually gets translated into, well, action, into what the cell does.
Right, and this leads to a really important distinction we need to make. Genotype versus phenotype.
Okay, what's the difference?
The genotype is the organism's complete set of genes, all its DNA. It's the potential.
The blueprint.
The blueprint, exactly. The phenotype, on the other hand, is the actual observable characteristics that result from expressing those genes. What the organism looks like, what it can do.
Can you give an example?
Sure. If E. coli has the gene called CEPMEX that's part of its genotype, its phenotype might be the ability to produce the Shiga toxin protein. The gene is the instruction. The toxin production is the observable result. Most phenotypes boil down to the proteins the organism makes.
So for a typical bacterium, what does this blueprint, this chromosome actually look like? Is it like ours?
Usually quite different. Bacteria typically have a single circular chromosome, and it's made of DNA, but it's supercoiled, wound up incredibly tightly.
Oh, tightly.
Get this. The E. coli chromosome has about 4.6 million base pairs. If you stretched it out, it would be about a millimeter long.
A millimeter. But the cell is microscopic.
Exactly. It's about 1,000 times longer than the cell itself. That's why the supercoiling is so essential, just to pack it all in.
That's amazing. You also hear about DNA fingerprinting.
Right. Scientists can use specific repeating sequences within that DNA called short tandem repeats, or STRs, to identify particular strains or individuals. It's a tool used in forensics, but also in tracking bacterial outbreaks.
And this whole field of looking at the entire genetic code is genomics, right?
Yes, genomics is the sequencing and molecular analysis of entire genomes. It's exploded in recent years.
How is it used in practice?
Oh, in countless ways. Clinically, think about tracking viruses like Zika or even COVID-19. Researchers sequence viral genomes from different patients and locations.
To see how it's spreading.
Exactly. By looking at tiny mutations, these little changes in the genetic sequence, they can build a family tree for the virus, understand its transmission routes, and even track how it might be evolving, maybe becoming more infectious or vaccine resistant. Genomics gives us a powerful lens on microbial evolution in action.
Okay, so we have the blueprint, the genome. Now, how does the cell actually use that blueprint to function and, crucially, pass it on? Let's talk replication.
Right, so passing information down through generations, that's vertical gene transfer. And the core process is DNA replication.
Which you said is incredibly accurate. How does it work? It's semi-conservative.
Yes, semi-conservative. That means when the double helix replicates, you end up with two identical DNA molecules. Each new molecule has one of the original parental strands and one brand new strand.
Like keeping half the old instructions and writing a new matching half.
Precisely. The process starts with enzymes relaxing the supercoiling and unwinding the double helix at a specific point, creating a replication fork.
Opening it up.
Then free-floating nucleotides in the cell pair up with the exposed bases on each template strand, A with T, C with G.
And the key player is DNA polymerase.
That's the master builder. DNA polymerase synthesizes the new DNA strands, adding nucleotides one by one following the template, and it's also a proofreader.
It checks its work.
It does. It has this amazing ability to recognize and correct errors, which is why the mistake rate is incredibly low. Maybe one error in every 10 billion bases added.
Wow. And there's a difference between how the two strands are copied, leading and lagging.
Yes, because the two strands run in opposite directions, and DNA polymerase can only build in one direction, 5' to 3'. So one strand, the leading strand, is synthesized continuously.
Smoothly.
But the other strand, the lagging strand, has to be made discontinuously in small chunks called Okazaki fragments.
Bit by bit.
Exactly. And then another enzyme, DNA ligase, comes along and stitches those fragments together into a complete strand. In bacteria like E. coli, this whole process happens bidirectionally, starting at one origin and moving out in both directions around the circle. Very efficient.
Okay, so the cell makes perfect copies of its DNA. How does it then use that DNA information to build the proteins it needs?
That involves two main steps, transcription and translation. First up is transcription.
DNA to RNA.
Right. An enzyme called RNA polymerase makes an RNA copy of a gene from the DNA template. It binds to the DNA at a specific start sequence called the promoter.
Like a signpost saying start copying here.
Pretty much. And it zips along the DNA, making an RNA strand that's complementary to the DNA until it hits a stop signal, the terminator.
And is the RNA copy identical to the DNA segment?
Almost. The key difference is that RNA uses the base uracil, U, instead of thymine, T. So where the DNA has an adenine, the RNA copy will have a uracil.
Got it. And this process makes different kinds of RNA.
Yes. The main one we talk about for protein synthesis is messenger RNA, mRNA. It literally carries the genetic message from the DNA out to the ribosomes where proteins are made.
All right.
There's also ribosomal RNA, rRNA, which is a structural component of the ribosomes themselves, and transfer RNA, tRNA, which plays a crucial role in the next step, translation.
Which is RNA to protein, the actual building process.
Exactly. Translation is where the cell machinery reads the message encoded in the mRNA and uses it to assemble a specific sequence of amino acids into a protein.
How is the message read?
The mRNA is read in groups of 3 nucleotides called codons. Each codon specifies one particular amino acid or sometimes a start or stop signal. This whole system is the genetic code.
And you mentioned earlier this code has degeneracy.
Yes, which is a fascinating feature. Most amino acids are coded for by more than one codon. Think of it like synonyms in language.
So a typo in the DNA, a mutation, might not actually change the resulting amino acid.
Correct. It provides a buffer, a kind of safety net against mutations causing problems. Some mutations end up being silent because of this degeneracy.
Clever. Are there specific codons for starting and stopping?
Absolutely. There's usually one specific start codon, AUG, which signals where protein synthesis should begin. And there are three stop codons, also called nonsense codons, UAA, UAG, UGA, that signal the end of the protein chain. All the other codons are sense codons specifying amino acids.
And how do the amino acids actually get brought to the ribosome to match the mRNA codons? That's tRNA.
That's the job of transfer RNA, tRNA. Each tRNA molecule has two important sites. One end carries a specific amino acid. The other end has a sequence of three bases called the anticodon.
Which matches the codon on the mRNA.
Precisely. The anticodon on the tRNA base pairs with its complementary codon on the mRNA. This ensures that the correct amino acid is brought into position according to the mRNA sequence.
And all this happens where?
On the ribosomes. You can think of ribosomes as the protein synthesis factories. They move along the mRNA molecule, reading the codons, facilitating the binding of the correct tRNAs, and catalyzing the formation of peptide bonds between the amino acids, linking them together into a growing polypeptide chain.
It's like a molecular assembly line.
A very sophisticated one. And there's a neat difference here between prokaryotes, like bacteria, and eukaryotes, like us.
What's that?
In bacteria, because there's no nucleus separating the DNA from the ribosomes, transcription and translation can happen at the same time. As the mRNA molecule is being transcribed from the DNA, ribosomes can jump onto the beginning of it and start translating it into protein immediately.
Wow, super efficient.
Very. In eukaryotes, it's more compartmentalized. Transcription happens inside the nucleus. Then the mRNA has to be processed. Non-coding sections called introns are snipped out, and the coding sections, exons, are spliced together.
RNA editing.
Right. Only after this processing and being exported from the nucleus can the mRNA be translated by ribosomes in the cytoplasm. It's a more complex, multi-step process.
Okay, so cells can make proteins, but they don't need every protein all the time. How do they control this? How do they regulate gene expression?
Regulation is key. Making proteins costs a lot of energy, so cells are generally very careful about only producing proteins when and where they're needed.
Makes sense. Are some genes always on?
Yes, a good chunk of genes, maybe 60–80%, are constitutive. These code for enzymes and proteins needed for essential everyday functions like glycolysis. They're basically always turned on.
But others are switched on and off.
Exactly. Many genes are regulated, often at the level of transcription, controlling whether or not an mRNA copy is even made. This is called pre-transcriptional control.
And the classic model for this in bacteria is the operon.
Yes, the operon model, proposed by Jacob and Monod. An operon is basically a functional unit of DNA containing a cluster of genes that work together, plus the control regions, the promoter, and the operator that regulate their transcription.
Can you walk us through an example, maybe the lac operon?
Sure. The lac operon contains genes that code for enzymes needed to break down lactose, the sugar in milk. This is an inducible operon, meaning it's normally turned off.
How is it kept off?
There's a separate regulatory gene that produces a repressor protein. This repressor protein binds to a specific DNA sequence within the operon called the operator site, which physically blocks RNA polymerase from accessing the promoter and transcribing the genes.
So it's like a roadblock.
A molecular roadblock, exactly.
Yeah.
But if lactose is present in the environment, or rather a derivative called allolactose, acts as an inducer. It binds to the repressor protein, changes its shape, and makes it fall off the operator DNA.
Roadblock removed.
Right. Now, RNA polymerase can bind to the promoter and transcribe the genes needed to metabolize the lactose. The presence of the substrate induces the expression of the genes needed to break it down.
Okay, so inducible means normally off, turned on by an inducer. What about repressible operons?
They work the opposite way. Repressible operons, like the tryptophan operon, which makes enzymes for synthesizing the amino acid tryptophan, are normally turned on.
Why would you want to turn them off?
If the cell already has plenty of the end product, in this case, tryptophan. If tryptophan levels get too high, the excess tryptophan itself acts as a corepressor.
It helps the repressor.
Yes. Tryptophan binds to the otherwise inactive repressor protein, activating it. This active repressor-corepressor complex then binds to the operator, blocking transcription and shutting down the pathway for making more tryptophan. It's a feedback inhibition loop at the genetic level.
Very elegant. Now, you also mentioned catabolite repression related to glucose.
Yes, the glucose effect. This adds another layer of control, particularly for operons involved in metabolizing alternative sugars like lactose. Basically, bacteria prefer to use glucose if it's available. It's the easiest sugar to metabolize.
So even if lactose is present, if glucose is also around, the cell might not bother turning on the lac operon much.
Exactly. The mechanism involves a signaling molecule called cyclic AMP, or CAN-MP. When glucose levels are low, CMP levels inside the cell go up.
Okay.
This CAMP binds to an activator protein called CAP, catabolic activator protein. The CAMP-CAP complex then binds to a site near the LAC promoter and essentially acts like a turbocharger for transcription. It helps RNA polymerase bind more effectively.
So high CAN-MP means low glucose, which means full speed ahead for the lac operon if lactose is also present to remove the repressor.
You've got it. For maximum expression of the lac operon, you need two conditions. Lactose must be present to remove the repressor, and D-glucose must be absent, so CAMP levels are high, activating CAP. It ensures the cell uses its preferred energy source first.
Beyond these operons, are there other ways genes get switched on or off? What about epigenetic control?
Yes, epigenetics is fascinating. This involves modifying the DNA itself, or the proteins associated with it, without changing the actual DNA sequence. A common mechanism is methylation, adding a methyl group to certain DNA bases, usually cytosine.
And this methylation can turn genes off.
It often does. Methylated genes are typically less likely to be transcribed. What's really interesting is that these methylation patterns can sometimes be inherited by daughter cells when the cell divides.
So even with identical DNA sequences, cells can have different gene expression patterns passed down.
Right. It helps explain phenomena like phase variation in bacteria, or why cells in a biofilm might behave differently from free-living cells even though they have the same genes. It adds another layer of heritable variation.
And control can happen even after the mRNA is made. Post-transcriptional control.
Definitely. One mechanism involves riboswitches. These are actually parts of the mRNA molecule itself, often in the untranslated regions.
The mRNA controls itself.
In a way, yes. These riboswitches can bind directly to small molecules, like a metabolite. When the metabolite binds, the riboswitch changes its 3D shape, and this change can affect whether translation of the mRNA starts or stops.
Well, what else?
Then there are microRNAs or miRNAs. These are tiny RNA molecules, only about 22 nucleotides long.
Tiny regulators.
Very powerful ones. miRNAs work by base pairing with complementary sequences on target mRNA molecules. This binding can block translation, or more often, leads to the enzymatic destruction of the mRNA.
So they silence genes after they've been transcribed.
Exactly. In humans, miRNAs are incredibly important for differentiating cell types, making sure a heart cell makes heart proteins and a skin cell makes skin proteins, even though they share the same genome. In bacteria, they seem to be important for responding quickly to environmental stresses like oxidative damage or nutrient limitation by shutting down specific protein production.
Okay, we've covered the blueprint, how it's copied, expressed, and regulated. But microbes are masters of change, of evolution. Let's talk about genetic change and gene sharing.
Yes, this is where the dynamism really comes in. Genetic variation is the raw material for evolution, and in microbes, it comes primarily from two sources: mutations and horizontal gene transfer.
Let's start with mutations. These are permanent changes in the DNA sequence itself, right?
Correct. A mutation is any heritable change in the base sequence of DNA. They can be neutral, having no effect, or they can be disadvantageous or occasionally beneficial, providing some new trait that helps the organism survive or reproduce better.
What kinds of mutations are there?
One common type is a base substitution or point mutation, where just a single DNA base is swapped for another.
Like a typo.
Exactly. Depending on the change and where it occurs, this can have different outcomes. A silent mutation doesn't change the amino acid sequence at all, thanks to that degenerate genetic code.
Yeah.
A missense mutation changes one amino acid for another. This might have little effect, or it could drastically alter the protein's function. Think of sickle cell anemia, caused by a single base change leading to one amino acid substitution in hemoglobin.
And a nonsense mutation.
That's when the base change creates a premature stop codon, UAG, UAA, or UGA. This usually leads to a shortened, truncated protein that's almost always non-functional.
What about adding or removing bases? Frameshift mutations.
Those are usually much more disruptive. A frameshift happens if you insert or delete one or two nucleotide pairs, or any number not divisible by 3.
Why is that so bad?
Because the genetic code is read in triplets, codons. Adding or deleting bases shifts that reading frame for everything downstream of the mutation. It's like taking a sentence, the fat cat ate the rat, and deleting the first THF, ATC, ATA, tetherat, complete gibberish after the change.
So it scrambles the entire rest of the protein sequence.
Pretty much. Frameshift mutations almost always result in a long stretch of altered amino acids and often hit a premature stop codon 2, leading to an inactive protein.
Do these mutations just happen randomly?
Some do. Spontaneous mutations occur naturally, usually due to errors made by DNA polymerase during replication. But the enzyme's proofreading function keeps this rate very low, maybe one mistake per billion base pairs replicated.
But the rate can increase due to mutagens.
Yes. Mutagens are external agents, physical or chemical, that can cause mutations.
Like what?
Chemical mutagens include things like nitrous acid, which can chemically alter bases, causing them to mispair during replication. Another type is nucleoside analogs, molecules that look like normal DNA bases but aren't quite right. They get incorporated into DNA and cause errors during copying. AZT, the anti-HIV drug, is an example. It interferes with viral DNA synthesis.
What about chemicals that cause frame shifts?
Some chemicals, like benzopyrene found in smoke and soot, or aflatoxin produced by mold, are potent frameshift mutagens. They can actually insert themselves between the stacked bases in the DNA helix, causing bumps that lead to insertions or dilutions during replication. Many of these are also carcinogens.
And radiation?
Radiation is another major mutagen. Ionizing radiation, like X-rays and gamma rays, has enough energy to knock electrons off atoms, creating ions and damaging free radicals. These can directly damage DNA by oxidizing bases or even breaking the DNA backbone.
And UV light from the sun.
UV light, especially UVB, causes a specific type of damage: thymine dimers. It causes adjacent thymine bases on the same DNA strand to covalently bond together. This dimer kinks the DNA helix and blocks proper replication and transcription.
So if DNA gets damaged, can the cell fix it?
Often, yes. Cells have evolved sophisticated DNA repair mechanisms. For instance, some enzymes called photolyses can use energy from visible light to directly break apart thymine dimers, reversing the damage.
Light repair.
Exactly. Another very important system is nucleotide excision repair. This involves enzymes that recognize the distortion caused by damage, like a thymine dimer, cut out the damaged segment of the DNA strand, and then use the undamaged complementary strand as a template for DNA polymerase to synthesize the correct sequence. DNA ligase then seals the gap.
Clever. It uses the backup copy on the other strand.
Precisely. That's the beauty of the double helix again.
Okay, so mutations happen and they get repaired. How do scientists actually find or select for mutants in the lab? Or test chemicals for mutagenicity, like in the case of Marcel Dubois, and potential carcinogens in food?
There are a couple of main strategies for finding mutants. Positive or direct selection is easiest. You set up conditions where only the mutant you're looking for can grow. For example, if you want penicillin-resistant mutants, you plate bacteria on media containing penicillin. Only the resistant mutants will survive and form colonies.
Simple enough. What about mutants that lose a function?
That requires negative or indirect selection. This is used to find, for example, auxotrophs mutants that have lost the ability to synthesize a particular nutrient, say the amino acid histidine, and now require it to be supplied in their growth medium.
How do you find something that can't grow?
You use a technique called replica plating. You grow bacteria on a master plate containing the required nutrient, like histidine. Then you press a sterile velvet pad onto the master plate, picking up cells from each colony, and transfer this pattern onto two new plates, one with the nutrient and one without it.
Ah, so the auxotrophs will grow on the plate with histidine, but not on the plate without it.
Exactly. By comparing the two replica plates, you can identify the colonies on the master plate that failed to grow on the minimal medium. Those are your auxotrophs.
And how does this relate to testing chemicals for causing cancer, like with the Ames test?
The Ames test is a brilliant application of microbial genetics to screen for potential carcinogens. It uses an auxotrophic strain of Salmonella that requires histidine, because of a mutation.
The same kind of mutant.
Yes. The principle is to look for reversion mutations that reverse the original mutation, allowing the bacteria to make their own histidine again, His+. The test measures how frequently a chemical causes these reversion mutations.
So more revertins means the chemical is more mutagenic.
Correct.
Yeah.
You expose the His Salmonella mutants to the chemical you're testing, often mixed with rat liver extract because some chemicals only become mutagenic after being metabolized. You then plate the bacteria on a medium lacking histidine.
only the revertants, the His plus cells, will grow into colonies.
Precisely. The number of colonies that appear is proportional to the mutagenic potency of the chemical. Since many mutagens are also carcinogens, the Ames test is a rapid and cost-effective initial screen for potential cancer-causing agents. It helps identify chemicals, like maybe those aromatic amines from heavily cooked meat implicated in Marcel Dubois' case, that warrant further investigation.
That's a really practical link. Now, beyond mutations within a cell, you mentioned microbes share genes between cells, horizontal gene transfer.
Yes, and this is a huge deal in bacterial evolution and adaptation. While vertical transfer passes genes down generations, horizontal transfer moves genes laterally between contemporaries.
And this involves genetic recombination.
Often, yes. Genetic recombination is the physical exchange of DNA segments between two different DNA molecules. When donor DNA enters a recipient cell via horizontal transfer, recombination often integrates that donor DNA into the recipient's chromosome or plasmids, creating a new combination of genes, a recombinant cell.
Why is this so important?
It allows for much faster acquisition of new traits than relying solely on spontaneous mutation. Think antibiotic resistance, toxin production, new metabolic capabilities. These can spread rapidly through a population via horizontal gene transfer. It's how Salmonella can switch its flagellar proteins to evade the immune system, for example, by recombining different flagellin genes.
And what are the vehicles for this transfer? You mentioned mobile genetic elements.
Right. Two key players are plasmids and transposons.
Let's talk plasmids first. Small, circular DNA.
Usually, yes. Plasmids are self-replicating DNA molecules, typically much smaller than the chromosome. They don't carry essential genes for basic survival, but they often carry genes that provide an advantage in certain environments.
Like what kind of advantages?
All sorts. There are conjugative plasmids, like the F factor, needed for one type of gene transfer. Dissimilation plasmids carry genes for unusual metabolic pathways, like enzymes to break down herbicides or pollutants useful for bioremediation. Some plasmids carry genes for toxins, like those causing diarrhea from certain E. coli or skin peeling from Staphylococcus aureus. Others carry genes for bacteriocins, proteins that kill competing bacteria.
And the really concerning ones medically are the resistance factors, or R factors.
Definitely. R factors are plasmids that carry genes conferring resistance to antibiotics, and sometimes heavy metals, too. They are a major reason for the spread of antibiotic resistance.
How do they spread so easily?
Many R factors are conjugative, meaning they can transfer themselves from one bacterium to another, even between different species. And the widespread use and misuse of antibiotics creates strong selective pressure bacteria carrying R factors survive, while susceptible ones die off. This drives the proliferation and spread of resistance. Think about our gut microbiome antibiotic use can kill off sensitive bacteria, allowing resistant ones, maybe carrying R factors, to flourish and potentially even transfer those resistance genes to incoming pathogens.
Scary stuff. What about transposons? Jumping genes.
Transposons are segments of DNA that can move from one location to another within a cell's genome. They can jump from chromosome to plasmid, plasmid to chromosome, or within the same molecule.
How do they jump?
They contain genes that code for an enzyme called transposase, which basically cuts the transposon out of its original location and inserts it into a new target DNA site. Simple transposons just carry the transposase gene.
But there are complex ones.
Yes, complex transposons carry other genes in addition to those needed for transposition. And very often, these extra genes are things like antibiotic resistance genes.
So transposons can move resistance genes around.
Absolutely. They are major players in spreading resistance genes between plasmids and chromosomes. For example, the gene conferring resistance to vancomycin, a powerful antibiotic, spread from Enterococcus faecalis to Staphylococcus aureus, likely via a transposon carried on a plasmid. They are powerful agents of genetic shuffling and evolution.
Okay, so we have these mobile elements. What are the actual mechanisms microbes use to transfer DNA horizontally?
There are three main mechanisms known in bacteria: transformation, conjugation, and transduction.
Let's take them one by one. Transformation.
Transformation is the uptake of naked DNA fragments released from dead cells directly from the environment by a recipient cell.
Naked DNA just floating around.
Yes. This was actually the first mechanism discovered, famously demonstrated by Frederick Griffith back in 1928 with Streptococcus pneumonia. He showed that something from heat-killed virulent bacteria could transform live, harmless bacteria into killers. Later work by Avery, McLeod, and McCarty proved that something was DNA.
Does any bacterium pick up DNA?
No, the recipient cell has to be in a specific physiological state called competence, where its cell wall and membrane are altered to allow DNA entry. Competence can be induced naturally under certain conditions or artificially in the lab.
Okay. Second mechanism, conjugation.
Conjugation requires direct physical contact between the donor and recipient cells. It's often mediated by a plasmid, specifically a conjugative plasmid like the F factor, fertility factor, in E. coli.
How do they make contact?
In gram-negative bacteria like E. coli, the donor cell, called F+, has genes on the F factor that code for sex polyappendages that reach out and attach to a recipient cell, pulling them together. In gram-positives, it often involves sticky surface molecules that cause cells to clump together.
And then the DNA is transferred.
Yes. A copy of the F factor plasmid is transferred from the F plus donor to the F recipient through a mating bridge. The recipient then becomes F plus F.
So it just transfers the plasmid.
Usually. However, sometimes the F factor can integrate itself into the bacterial chromosome. Such a cell is called an HFR cell, high frequency of recombination.
Why high frequency?
Because when an HFR cell tries to conjugate and transfer the F factor, it starts transferring the chromosome itself, beginning at the integrated F factor site. The connection usually breaks before the entire chromosome is transferred, but significant chunks of chromosomal DNA can be moved to the recipient, where they can recombine. This was historically used to map the order of genes on bacterial chromosomes.
Clever. And the third mechanism, transduction.
Transduction involves bacterial viruses called bacteriophages or just phages. These viruses infect bacteria, replicate inside them, and sometimes during the assembly of new virus particles, they accidentally package a piece of the host bacterium's DNA instead of or along with their own viral DNA.
So the virus particle carries bacterial DNA.
Exactly. When this faulty phage particle infects a new bacterium, it injects the bacterial DNA it's carrying from the previous host. This donor DNA can then recombine with the new host's chromosome.
Does it transfer random DNA?
In generalized transduction, yes. Any piece of the bacterial chromosome has a small chance of being packaged into a phage head. But there's also specialized transduction.
How's it different?
In specialized transduction, only specific bacterial genes, those located adjacent to the site where a temperate phage integrates into the host chromosome, are transferred. This is actually how some pathogenic bacteria acquired their toxin genes. For example, the phage that carries the diphtheria toxin gene for Corynebacterium diphtheria, or the Shiga toxin gene for E. coli O157H7, transfers these specific genes via specialized transduction.
So viruses can actually make bacteria more dangerous.
They absolutely can by acting as vectors for horizontal gene transfer, particularly for virulence factors like toxins.
So if we pull this all together, mutations create variation, recombination shuffles it, and horizontal gene transfer spreads it around. This is the engine of microbial evolution.
That's a perfect summary. These genetic mechanisms constantly generate diversity within microbial populations.
And then natural selection steps in.
Exactly. The environment, whether it's the human body, the soil, or a hospital surface treated with antibiotics, acts as the selective pressure. Organisms with genotypes that confer an advantage in that specific environment are more likely to survive and reproduce, passing those advantageous genes on.
Like antibiotic resistance.
Antibiotic resistance is the textbook example of natural selection in action, dramatically accelerated by human activity. Our widespread use of antibiotics selects strongly for resistant strains. Microbes are constantly changing, adapting, evolving in response to their surroundings, all driven by these fundamental genetic processes.
And that feels like a good place to wrap up this deep dive. We've really covered a lot of ground, from the very basic structure of DNA
to the complex ways genes are regulated, swummed on and off,
and the dynamic ways microbes mutate and share genetic information, driving their incredible adaptability and evolution.
Yeah, understanding microbial genetics, it really isn't just an academic exercise, is it? It's fundamental to tackling infectious diseases, developing new biotechnologies, managing our own microbiomes. It's central to so much.
It makes you wonder, what breakthroughs are next? What new insights will decoding these tiny blueprints unlock for us in the future?
It's constantly evolving. New sequencing technologies, new understandings of gene regulation. It's a really exciting field.
Well, thank you for joining us on this exploration into the world of microbial genetics. We hope you, our listeners, feel a bit more informed, maybe even a little more curious about the microscopic world buzzing all around us and within us. Thanks for being part of our last-minute lecture family.